let me re-emphasize. It is a lot of information that we are giving you here. I guess even too much information, and I totally get it if some of you feel a bit overwhelmed, even frustrated at times, that I don't get it, why is it not working for me? And I will never learn this. This is how I felt when I first started programming. It is way too complex. It's not way too complex. It's just a lot of information crammed into 2 weeks, and it's impossible to learn this all. And that's why I said, get your basics right. Know how to use R, know how to use Linux. Because once you know these two tools, you can basically apply everything. The rest is always the same. You always use functions. You know how to use functions. You know how to get something back from a function. You know how to use your Linux commands. You know how to look for help in Linux. You should know how to use Google. Google is your friend. You should know how to use Stack Overflow. It's very unlikely that any of you, you're in the middle of your career. You are already, most of you have their first degrees. Some of you have second and third degrees already. It's very unlikely that you will become a bioinformatician who develops advanced programs. This is usually done by people who have PhDs in maths and computer science. So this will not be your major objective for the future, I guess. You want to use these tools so you can pursue your research in plant science, right? And that means all these tools have already been developed. They're there. You just need to know how to use them. Okay, so this is something I wanted to emphasize. This brings me back to weighted gene co-expression network analysis. When I was in the lab of Steve Horwath at UCLA, who developed this method, he has a background in, he has his first and second degree in maths, and he did the PhD in biostatistics. So he's a mathematician, okay? This is how he developed this method. He's not really a biologist. When I talked to him about biology, he was a bit lost. And he had his programmer, Peter Langfelder, who actually wrote this WGCNA, who did all the programming. So he developed the math and had a programmer who programmed it. Okay? Just for you to know, and don't get frustrated. Yeah. So yesterday we all installed WGCNA. You see I have 2 tutorials for WGCNA, since it's quite a broad subject. And in each tutorial, you will have 6, first tutorial will have 7 files, 7 R files, and you have the PDFs, you have the data. Here in Team and Liver, you will have the data. And in the second tutorial, you have also 7 files. 1, 2, okay. I don't think that we will manage to go over all of them. So we will start with the first one. And once you get the idea, you can go back home and finish them all by yourself, get experts, become experts in weighted gene co-expression network analysis, and then apply it to your data sets. If you have RNA-seq, so once you have RNA-seq data, you do what Juan taught you yesterday. So you do what Juan taught you yesterday. You get all the counts. Once you have your counts, you normalize your counts. You can do this with this package. Can you do it with HR, the normalization of the counts? Yeah, so you have these packages, HR. You have the packages DE-seq in R, where you get your data and you can normalize your counts. There are tutorials for that too. You just have to follow each step by step, and then you will get your counts. You can use only the differential expressed genes to do WGCNA. That means if you believe in the statistics of differential gene expression, you only extract the genes that are differentially expressed. Why do you laugh? If you believe in it, yeah. So you only extract those, and you only work on those. The other ones you can throw away. And then you use the differentially expressed genes to run WGCNA. Okay. Once you have your normalized counts, you start WGCNA. So here's the tutorial. Okay. I will do this. You can follow me, or you can do it by yourself. You don't have the files. I gave them to you. Bad monkey. I will give it to you. No more worries. Bad David. Sorry. Next time you hit me. Someone has them, or none of you have them? Salute. But you have the lecture, right? That we used yesterday? Yeah. Start one here, start one there, and one from the back. So the two folders, WGCNA1 and WGCNA2. And it's based on a microarray data set, but it doesn't matter. In a microarray, you have a set number of genes. Whereas when you do RNA-seq, you have the entire transcriptome of the sample that you're looking at, and you potentially could find all the genes of a species, transcribed genes. So now I'm in my WGCNA1 tutorial 1 folder, and you have all these files. You have the PDFs, and you also have the R scripts. So if you want to cheat yourself, you just open the R script. If you want to learn the method, you open the PDF file. Okay. So what I will do, what I always do, I open a new text editor, because I like to see, you can put this all in history, but I like to see what I'm doing, so I'm saving each step, each command in the procedure of my of scripting. So it is suggested that you first see where you are, of course, so you can get the data right. So we start with get WGDD. So I'm in the wrong. A good practice is to have a sort of description at the beginning of your file. Okay? Let's say you put your date. You put today's date is March 14th, 2019. So when you go back, you know exactly when you did what at what date. So no one can steal your data. You put author, your name, not your name. Short description. So instead of changing my working directory in code, I can just go to my RStudio and click on more. I navigate to my folder where I want to be. Yeah, here, and just click on the folder. I say more, and set as working directory, and then I just copy the code so I know what I did. So I know where I was working at. So here I immediately know, this is the my working directory. Okay, if you get the same error as I have, so I don't want to read in as factor. The factor is if you run an ANOVA, everyone knows what the an analysis of variance is. You have different factors by which you make the computer understand what to look for when you look for analysis of variance. And we don't want this here. We don't want the strings that we read in to be considered as factors by R. So we set this option to false. Okay, now we're reading the data. So we start with the female mice. And there's a function called read CSV. Instead of using the function read table, I can just say read CSV, and my separator will be a comma. CSV stands for comma separated values. If I just put the dot and a slash and tap, it will automatically show me all the files. And the data is in the folder female liver data. And here's my liver female. Reading it in. Okay? I'm checking. I can just look. If I look at head, it's not such a good idea because the data set is rather big. So first I'm checking the dimension of my data file. So it shows me I have 3,600 rows and 143 columns. Right, okay? If I want to check the names, so these are all the different samples. If I just want to look at the data, I wouldn't recommend to do head. So what I would recommend to do is just looking at the first 10 rows and the first 5 columns. And what you will see here is you will have a lot of data, a lot of columns that you don't want to use. Some of them will give you just description of your files, description of what has been done, reference and so on and so forth. So we need to get rid of this data. So we need to clean the data first. And this is done by the first line. So what I'm doing here is I'm deleting the first 8 columns. Can you see that? Okay. If I can, so T stands for transpose. So instead of wanting to have my rows, my genes in rows, I will transpose my data frame. Okay, transpose it by 90 degrees. And so I will have my genes as columns and the sample names as rows. This is how WGCNA needs the data. You need to have the genes in columns. Because right later on when we correlated, the core function of R for correlation correlates columns to each other, not rows. That's why we need to have the genes in columns. And since I have this sort of descriptive data in my first 8 columns, I'm deleting those. If I put a minus C1 to 8, it will delete these first 8 rows, 8 columns. Yeah? Done. So if I look at the data again, so you can, just not to confuse you, you can use an equal sign, but I prefer using the assignment sign, which is this arrow. But they will do the same thing. Okay? Now, I have my clean data in the variable that express 0. We'll look at it again. First dimensions. And now you will see, first I have 135 rows, right? and 3,600 columns. If I do this with my original data set, I will have first 3,600 rows and then 143 columns. Since I transposed my data and deleted 8 columns, I will only have 135 rows left. Okay, do you see that? Right? 143 minus 8 is 135. But we have the same number of genes. Can check the names now. I'm going to look at the beginning, and these are all the names. Let's check the row names instead. And what you would see here is that I don't have these 8 column names that I had before. They are gone. You follow me? Good. No? No? Too fast? Okay, where are you stuck? Where? On Mars? I said that. If you get an error message while loading the package, it will tell you you don't have a package for GoTerm analysis. Since I don't believe in GoTerm analysis, we won't do GoTerm analysis, and you can ignore this error, okay? But your package WGCNA should be loaded. The warning. And some of you, as here, you will get a warning. This package was built under this and this and this version. It's just a warning. As long as you don't get an error in R, and you just get a warning, it's just R telling you there might be something fishy here. But most of the times, 90% of the times, you can ignore those warnings. Sometimes it will tell you you get something, but it's not what you wanted. So in that case, you shouldn't ignore the warning. Data that I cannot, this is just descriptive data. If you look at the first 8 columns in the original file, FEM-data, okay, just what I did here, FEM-data 1 to 10, 1 to 5, you would see that there is information which is only descriptive. Yeah, these are numbers that you cannot use to do, these are not quantitative numbers for your gene expression. These are descriptive numbers, and we cannot work with them. We need to delete those. And this is what we're doing. This is what we're doing here. We're deleting the first 8 columns because we don't need them. And then we're storing it in the variable .express0. Sometimes you don't have an option but to actually look at the data. You can open them in an Excel file, and it will show you this is data. Usually when you run something, let's say in an RNA-seq or a mass spectrometry, they will give you descriptive data. This descriptive data is useful. Metadata, yeah, it's useful. Since you don't want to know what the origin of your data is, what the method that was used, who did it, what machine was used, and so on and so forth. But for the analysis itself, you cannot use it. So why are we removing these first 8 columns? The first of 8 columns are metadata, showing you the description of how your microarray was done, under what conditions, who did it, where was it done, and so on and so forth. But it doesn't give you gene expression data. And this is what we're basically interested in. We only want to have the expression values. This is one one step, if you get into bioinformatics, you will see this is a step that you will always have to do, because when you work with biologists, they don't understand that things have to be done in the same manner over and over again when you use computers. You saw yourself now in the last 10, 11 days that you have to be very precise when you type in your words, when you type in characters. If they are a bit different, if one character is lowercase, the other, and in the next one it's an uppercase, it will not work. For you as a human, it's obvious, but for the computer it isn't. So the cleaning data of the cleaning process of your data is all something that you will always need to do. You cannot rely on biologists to do it for you. There will always be mistakes, always. Even if you tell them exactly what to do, they will not follow your instructions. Okay, let's continue. Okay, the next function doesn't work because we don't have, because there's no, it's not available for the current version of R. So we will have to write our own code to get rid of columns that are only represented by zeros. Okay? How do we do this? Okay. We can use the function apply. We've seen that before in our data set. So X stands for the data set. Margin, you want to apply it either to rows or to columns. So the margin should be, if you look here, a vector given is for matrix 1 indicates rows, so I want to run it over all rows. I want to see and then sum. I want to sum up each row, each column. No, it should be column, sorry. I want to sum up each column. Since I have the genes ordered in columns, I want to sum up the value of each column. Okay? Okay, now I have the variable 0 rows. Let me check. The length of my variable is 3600. Yeah, if I look at it, I will get a vector that has 3600 entries, with each one with the sum of each column in my data set. Okay? Now I can check which of those is 0. Right? How can I check which entry in my vector 0 rows, which entries are 0? There are different ways to do it. I like to use the function which. Actually, it should be 0 calls. Sorry. And I will remove the variable 0 from my memory. Okay, so, right, the double equal sign is a conditional statement. Okay, which 0 calls are equals 0. And I will store it in a variable called x. If I look at x now, none of them. So I have for each gene, I have some entries. I have some expression value for each condition. Not for each condition, but at some conditions. Doesn't mean we'll have conditions, we will have expression values in all of them, but no gene has no expression value. Okay? So if I were to remove some of these columns now, I could say greater 0, right? We had if statements. If I know that the length of X, since I looked at it, is 0. But let's say I found a few of the genes that didn't have any expression values in none of my conditions. That means the length of X will be greater than 0. It will be as big as the number of columns it found. So I'm making a conditional statement. If the length of X is greater than 0, remove these columns. How do I do this? I use my data set and say minus X. The X will correspond to the indices of my columns, and I want to remove those. So so that you believe me that we don't remove anything, if I look at the dimension of my data set now, it's 135 rows by 3,600 columns. If I execute this thing, and I look at the dimension again, it is still 135 rows by 3,600 columns. Why? Exactly. I didn't remove anything. But if I were to remove, this is how you could do it. Okay? So all this part here, we can skip. So, right. Identifying outliers. What's going on? Okay, now, how well are you familiar with statistics? Basic statistics. If you are running a basic statistical test, you have to check, if you want to run a parametric statistical test, you have to check that you don't have any outliers in your data. How can you generate outliers? These outliers can be a mistake in the process while measuring the data. This can be something during the experiment that happened. Something that is an error while you were actually doing the analysis in the lab, and so on and so forth. So we want to make sure that we don't have any of these outliers, because if we keep those outliers in our data, we will get a bias. It will change the way our models will look. So we need to make sure we remove these outliers. And these outliers are obvious because they don't integrate themselves in the normal distribution of the other any other data that you have. So it's quite easy to identify those. There are different tests how you can do it. If you want to look it up, look up Cook's test. The Cook's test will identify outliers in your data set and will tell you remove this. You can do this also in R using a Cook's test. How we do this in WGCNA? So first, we build a tree, a sample tree, which clusters together, which uses hierarchical clustering to cluster together the data based on the expression values that we have. We didn't run a correlation analysis yet. We're just looking for outliers at the moment. Okay? If you enter sample tree in your console, it will tell you, it will give you some statistics. So the cluster method that we use is based on average. So we're averaging all of our data, and based on the average value that each gene gives us over the different conditions that we are checking, it will cluster them together. How many conditions do we have? 135. Or 135 samples, better said, because we have less conditions because we're working with replicates. That we're using in this hierarchical cluster method is Euclidean. Are you familiar with Euclidean distances? Euclidean distance basically is the distance between two objects that you have on a two-dimensional space. Yeah? We don't have, so if I'm plotting my two-dimensional space, this is my two-dimensional space. Imagine here I have two axes, x-axis and y-axis. I put a data point here, I put a data point there. The distance between those two is my Euclidean distance. So now we're going to plot the tree so we know what it looks like. Can you all see this? Okay? So, these are, you have the average values for each of the genes. No, it's not the average value. For each of the samples, and then they will be clustered together. And then you see something here. You see this? This is an outlier. It doesn't fit in with any of my data. So I will have to remove it. So make it a bit clearer, we can use the function addLine. Yeah? So everything beyond this line will be determined an outlier. It's very unlikely because it doesn't fit into the rest of the data. I have replicates. Okay? So if I have replicates and it doesn't conform with the other replicates, something went wrong with in this specific sample. Yeah? So we saw plotting before. What R allows me to do is I could plot, if I wanted to, as many plots next to each other as I wish. So with the function par, which is sort of the mother function of all plot, I can predetermine the plot that will come when I use the actual function plot. I can put here also number of rows and number of columns. If I wanted to plot 4 plots next to each other and actually save them, I could say number of rows 2 and number of columns 2. So we'll have a 2 by 2 plot, and I can put in, and each time then I use the plot function, I will first plot one here, plot one there, plot one there, and plot one there. But this is sort of the, in terms of hierarchy, this is the mother function of plot. So if you want to use some of the parameters that are available in plot, You need to look them up in the the help of path. Yeah? Let's go on. Ah, shoot. You can also correlate data that do not originate from the same experiment. But I wouldn't do that because this sort of publication can be rejected very easily, because you can have correlation by chance. Actually, there's a lot of people who actually run correlation just for fun of it and show and actually make a point that correlation is not causation. But you can just find correlations randomly, and they don't mean anything, right? Who showed this? Did you show this comic?
Yes.
Yeah, yeah, that, right. When we all shave our heads, then we will be more successful. This is just a random correlation, and there are many out there. So always. And this is also something, when you design your experiments, make sure you have enough sample size, samples and biomass, so that you can use them for gene expression analysis, for metabolic analysis, for proteomics. Or let's say you don't know what you're going to do, you just want to do gene expression analysis at the beginning, save some in your -80. So you can do it later on, if you need to. Yeah, so now we're going to import the clinical data. We have now expression data from mice liver, from female mice liver. And now we want to correlate this to clinical data of these female mice. We use the function again, read CSV, as it's stored as a CSV file. And then we can look at it. Now, instead of doing it in the console, I can also just go into my environment, click on it, and then you will see a sort of spreadsheet will show up. Okay? So here I have the sample numbers that correspond to the sample numbers that I had for my gene expression data. You have some number, I don't know what it means, some mouse ID, the strain of the mouse, the sex of the mouse, date of birth of the mouse, their parents, their diet, sac date, not sure what it means, their weight, their length, their AB fat, other fat, total fat, so all these things that were measured. Fat weight, the triglycerides, the cholesterol, HDL cholesterol, you see all glucose. So since we're looking at liver values, we want to know, we want to correlate it to some dietary traits. And this is what we're going to use. So again, we need to clean the data. Yeah? I will just copy all of this. We looked at the data now with our eyes, and we determined which of these columns are not useful to us. So we remove columns 31 and 16. And then we check the dimensions. So we have 361 rows and 27 columns. Now we look at the names. These are all the different traits that we actually want to correlate. And see whether we have correlation in to our genes. Yeah. So since we removed one of our samples from our data expression, from our gene expression data, we also need to remove the sample from our phenotypic data set. So this is how we're going to do it. So we have our female samples, right? These are all the row names that we have of our female samples. And you see there are 134. So there's a function match. So we can match the row names to the traits of mice. That should be the same. So if we just execute it without storing it, you will see my female sample F2_2 matches my trait sample in column mice at row 207. My sample F2_3 matches my sample in row 208 in my column mice in my phenotypic data. This is similar to the merge function that Juan showed you yesterday. But sometimes, if I were to do it the other way around, I would show you this. If I were to compare all traits of mice column, which holds these names, the same names, the sample names, to my female samples, what will you see? Lots of NAs. Why is that? What do you think that is? Look at the dimensions of all our traits. We have 361 rows and 27 columns. The rows correspond to my samples. How many different samples did we have in the gene expression data? We removed one sample. How many did we have before removing? 135, right? How many did we have after removing? 1. 134. How many do we have here? 361. What does this mean? Do we have the same number of samples that were subjected to gene expression analysis as we have the same number of samples that were measured their phenotypic traits? Yes or no? No, exactly. So for some reason, probably for financial reasons, they decided just to take 135 samples out of the 361 samples that they had for gene expression analysis. How do you do this? How do you determine which samples to take? So there are statistical analysis called multivariate analysis, something like principal component analysis. What I would do, I would take the phenotypic traits and take the samples that are the most apart from each other. The ones in the center, I would say they are very similar to each other. So by that method, I could determine which samples to choose. Let's say I only have a certain, my finances only allows me to do 135 gene expression analysis. So I have to determine somehow how to choose them. So now we have to see that we match those 135 samples, actually only 135 samples, to our phenotypic traits. So we are matching with the function match. The row names, here are my row names, female samples, to the row names, to the sample names that I have in my phenotypic trait data set. Yeah? If I execute this and look at the length of my vector trait rows, it will be only 134. So all the other 227 I couldn't find. So I will only work with the ones that I have in my gene expression data set and the ones that I also have that correspond to the corresponding ones that I have in my phenotypic data set. Yeah? Is this clear? Okay. Yeah, okay. So this is exactly what I'm going to do here in this row. Now, I have my trait data set, my reduced trait data set. Yeah, if I look at the dimensions of that trait, it's 134 by 26. So I have 134 samples. And this is what I want. To make sure that they have the same row names, I would just give them the same row names. This is what this line does. If I look at the row names, this is just to make sure. They should have the same row names anyways. Okay? Continue. Yeah, okay, what it says here. So now we have the expression data set and we have our trait data set. So we want to see how one corresponds to the other. So we recluster them. Remember that we removed one of the samples. So these numbers to colors will convert the numbers to colors according to the program of WGCNA. Until now we were able to somehow, whenever these errors occurred, we were able to get our way out of it. But now we will not be able to, because this is a essential function of WGCNA. Okay. So I fixed it. Yeah, the issue is we need to install some other packages on which WGCNA is dependent on. So I will put all the packages here. Install them, please. Yeah, those 4 packages, if you install them, then WGCNA should work. Yeah, this is going to, no, they're not so big. GoDB, yeah, except for GoDB, all of them are quite small, so. You never know. What else happens in Linux sometimes? Yeah. Yeah. No, and after that, run library WGCNA again, and then you shouldn't get any errors. Data imputation. I think at one point it will ask you whether to update the library matrix and the library cluster, if I'm not wrong. Type A, you have the options A, N, and Y. Type A for all, and then hit enter. What? You're right. Ah, yeah. You're right. Yeah, you're right. You could also just put everything in one line. Put a C, like bio C manager ::: install, open bracket, C, open bracket. The names GoDB, impute preprocess core, MVT norm, divided by commas, close bracket, version 3.8. And then it should install all of them. Or you can just do it like this. Do you start up? Exciting. What? Geo. That's a go. Hmm? Yeah. I don't. Idiot. I don't have an option. You know what it does? You can take those modular identifiers and then apply go terminology to it. So, like I said, I don't want to. You can talk. You can talk. You want to tell him something? No. I have an idea for a project.
What?
I have an idea for a research project.
Yep. Good.
No, for you.
Oh. What is it?
So, The fourth one is MVT, not MTV. Pre-processed, not pre-processed. My bad. Try again. Yeah. It's because so many people are using the internet, so that. In the meantime, while you're all installing this, I would like to show you a package to construct graphs in general and also to analyze them. It's called iGraph. Because I updated to this stupid version. So the package iGraph is another package just for graphs. It doesn't have to be biological graphs, but it gives you many options to analyze your graph. So let's say you construct a graph using WGCNA, then you just bring it over into the package iGraph, and then you can actually look at the graph in terms of graph properties. This is something that I showed you yesterday, right? Where you look at the degree of connectivity. You could look at edges and nodes and so on and so forth. So what you can always do just to explore a package, there you have, I have my iGraph package, okay? So the first thing I do is look at all the functions it provides. And you will see there are quite a lot of functions in this package. Okay? So let's say I want to construct a graph with iGraph. So no, that's not the one. So here. So the function graph from adjacency matrix. What I told you yesterday, if you have a symmetric matrix, an M by M matrix, you can, this is basically your network. Each connection between a row and a column and your, in the cell value displays your edge. Okay? So I will get the entire description here. And at the end of, usually at the end of the help file, it will give you code how to do stuff. Okay? So let's say I want to construct a matrix I just have to copy the code. So this is my graph. Yeah, I have a graph with 10 different nodes. And node 2 is connected to node 1. Node 2 is connected to node 4. Node 2 is connected to 10. Node 3 is connected to 2, and so on and so forth. Okay? Now I can use other functions from iGraph. Let's say I want to know exactly how many nodes, how many vertices, that's the V, what the V stands for, in my graph. 10. I can do the same count. Edges. I have 11 edges. I can print my graph or plot my graph. And look at it visually. So iGraph is a very, very extensive library to analyze graphs and networks. You can also find this library under Python if you wanted to. So every 1 minute to install all the packages. So I have a, no, doesn't work. Zena? I'm sorry. I just updated my R to the latest version prior to the course. Actually, was still working in version 3.4. And everything worked there, so. So. Based on the average values are associated with some of the samples. Okay. So now here's something called the collect garbage. A garbage collector in programming is basically something that frees up your space, your memory. Because sometimes you have so many variables that you don't see, but they are stored in your memory and you want to vacate the space. So they use the function garbage collector. How do you spell it? Collect garbage, sorry. So this is something just to make it, but you can just say GC, garbage collector, which is pre-installed into our base. And it tells you, gives you a message. This is the amount of memory you have available, the amount of memory that is occupied. again, be careful typing. That's the capital O, not lowercase o. If you put a lowercase o, it would give you an error. You cannot find that. Yeah. No. Okay. So now we can, it shows you to save the data. We will skip this. And this is, these are the different figures. Okay, this was the first tutorial, just to get your data started, just to get your data into your memory and to see how they cluster together. That's all. The second tutorial so this assumes that you saved your data, but since we didn't save the data, we will just continue. And it will actually ask you again to set your options to string as factors false. Salute. Here they have a new function called enable WGCNA threads, which basically tells you I can parallelize my data. Allowing parallel execution with up to 3 working processes. It's just to enable it, okay? We can skip this since we still have the data. So now is when we're actually starting to construct the network. And this is the first thing that we have to determine is the power. Remember yesterday when I showed you that you have to raise your correlation coefficient to the power of beta. So now we have to determine the soft threshold. How do we do this? Okay, so we create a variable called powers. So look at my variable. It's just a vector from 1 to 20, not more. Okay. Now, in this function, I will use my data expression, data set, and I will use powers. It will go into a loop and raise each matrix. It will raise the matrix to the power of 1, 2, 3, 4, 5, 6, 7, up to 20. And it will store it in the variable sft. Yeah? This new version is a nightmare. Don't do this. It will take longer. Okay? It was still rather fast. So don't enable parallel processing. Somehow, for some reason, it gives an error message. So I just disabled it again with the function disable WGCNA. This is quite new. Never saw this before, to be honest. Okay. Now, how do I know what my soft threshold should be. I plot it. I will just copy this up to here. This is what I mentioned before. Okay, so let's go over the rows. So my size, here I will determine the size of my window, 9 by 5. I will tell, before I plot, I will tell using the function par, I want to have 1 by 2. That means I want to have one row and 2 columns. Okay? Then I will plot my soft threshold plot, and then I will plot the connectivity, yeah, corresponding to the powers. So here is the line which determines when my network becomes scale free. Who remembers what scale free means from yesterday's lecture? What do we want to achieve? Do we want to have a network where we have all nodes connected to each other equally, or do we want to have a network where we have hubs in our network which are connected to many other nodes by strong correlations? The second one, we want to have a scale-free network where we have single hubs, and these hubs will be the main genes in our modules. Okay? And this happens at the threshold when I raise the network to the power of 7. And when I have 7 here, you will see it will become scale free. Remember this graph that we saw yesterday where we had different lines? One was a diagonal, and at 1 it started to bend a bit, at 2 it started to bend even more. This is exactly what this represents, just the other way around. Okay? So now I know the soft threshold power that I should use to construct my correlation network is 7. Okay? So this is what we've seen ourselves. We constructed this ourselves. Okay. Now we will construct. For some reason they use the power of 6 here. Yeah, 6, 7, same thing. So why do I say same thing? Don't be confused. Here, when I have 6 and 7, the networks are very, very similar. So whether you choose 6, 7, or 8 won't make a big difference. In any case, sometimes you will not get to this. It will even out here at 0.6. And then you will, when you plot the red line, you would see none of the powers that you get are even close to getting a scale-free network. But you should never go beyond 20. Because then correlation coefficients will be so little that you cannot use them anymore. It will not tell you anything about the network. So now I will construct my network using blockwise modules. What does it mean? Let me run it. I think it takes a while. Okay. Remember, I have a 0 behind it. Okay, so I have my data expression, my gene expression data, and I know that my soft threshold is 6. Okay? So now I'm using the correlation analysis. I'm making correlate all the genes to each other and raise the correlation coefficient to the power of 6. This is the distance matrix that I will have. It's the correlation itself. And I'm telling the computer, I want to have at least, if you now, it will now order the different genes into modules. And I'm telling the computer, the minimum module size should be 30. If there is a module less than, don't construct modules that have less than 30 genes. If there's a module that is a little bit outside, just take it in order to get 30 genes. So I'm reassigning threshold 0. I'm not using it. So merge. This corresponds to the module size. If the threshold of a module is less than 25 from one module to another, merge those modules together. Yeah? And save it. This is where I will save it. Ham, respect, dentro. Whew. So this is called partition around meteoroids. What does it mean? It is another clustering method. And how do I cluster? Instead of clustering around the means, I'm clustering around the median. And I don't want to do it. So I say false, because I already know that I want to use the average. Okay? It's done. Now I should have a new file, because I'm telling it save my blockwise construction into female mouse Tom, and a new file is generated down here. Okay? So this is what we have. And this is the automatic model that you can just say to the computer, do this. I'm not interfering. Just tell me what you think the best model should be. We can also do manual hybrid models, and we can do completely manual models. The way you usually go by is you make first the automatic model, then you make the manual hybrid, and then you make the manual one. You compare one to another. So if the first one compares well to the second and the third, the automatic compares well to the blockwise and also compares well to the manual one, you can use the automatic one. If the second one compares well to the first and the third, but this first doesn't compare well to the third, use the second one. If the third one compares well to the first, to the second, but the second only compares well to one, and also the first compares well to one, you use the third one. Yeah? Am I making myself clear? I doubt it. If I have a model, my automatic model, it compares well to my second model, my manual hybrid model, and it compares well to my manual model, I can use the first one. If my second one compares well to the first one, and also compares well to the third one, but my first one doesn't compare well to both, just to one of them, and also my third one doesn't compare well to the first and the second one, I use the second one. If the third one compares well to the first one and the second one, but the first doesn't compare well to the first, only to one of them, and the second also only to one of them, I use the third one. If all compare well to each other, I can use either one. You can compare. I know it sounds complicated. It's a bit confusing. It reminds me of another movie. You know the. Huh? Yeah, you basically need a decision tree for that. That's true. Okay, now we have our automatic model. So, there are these functions. Again, I want to plot it so you can actually see it. What does this find? Labels two colors. So and now I only have labels in my model. And each module is each gene is represented as a label associated with a certain module. In order for me to visualize where each gene belongs to, I'm converting these labels to colors. And that's all the function labels to colors does. Now I'm plotting it. Aha. Okay. Do you remember this dendrogram that we had before? It's the same one. What do you see here now? Colors? Right. What does it mean? It tells me to which module each gene belongs. No, I'm sorry, that's not the same dendrogram. We had a dendrogram of the samples, not the genes. So that's a dendrogram of the correlations of the genes. Yeah? This is this dendrogram is based on my correlation values raised to the power of 6. This is how they cluster together. You will see here, for instance, have very clearly all the genes belonging together. And you have this whole thing is module yellow. So these genes should belong together based on their expression values. Okay? Now here, you see something that doesn't look so nice. I have turquoise, I have pink, I have purple, I have a blue. So how can it be that this module or these genes belong to that gene, but we have another module in between? Because you did an automatic modeling. This is what the computer thinks. So now is where you need to intervene and tell the computer, merge this. If these modules are close to each other by their distance, merge these modules into one. Okay? And this is when you do the manual hybrid and the manual one. Okay? Sometimes it just comes out perfect. Really. So you can use the automatic one. In that case, the automatic one will also will fit with the manual one and the manual hybrid one. Okay? Okay, next thing. We're only looking at the automated network at the moment and the automated modules. Yesterday I told you in order for me to correlate my modules, which contain hundreds of genes, to my phenotypic data, I need to do something, which was I need to calculate my module eigengenes, which are my eigenvectors. Okay? This is based on linear algebra. So if you have a matrix, let's say we have a map of Lima. I'm putting it on the wall, and I'm putting it from south to north. I'm plotting an arrow from south to north. I'm plotting an arrow from east to west. And I'm plotting an arrow from southeast to northwest. From from southwest to northeast. Now I say I'm stretching my map. Someone holds it at the top, and someone holds it in the middle, in the bottom, and they stretch it. What will happen to these arrows? What will happen to the diagonals? They will change their conformation, right? Because you pull them, they will actually tilt it. What will happen for my east to west? It will get thicker, right? I'm pulling it apart. It will become thicker. What happens to my south to north? It will only become longer, but it will not change. It will get longer, but actually will not change. This is my eigengene. This is the one that represents my data in one single vector. If I was just to pull it, and I would just to extrapolate more values, I can use this vector to do so. This is linear algebra explained in a few terms, okay? And this is exactly what we're doing here. For each module, I will calculate now. Oh, sorry, that's the wrong one. Okay, so again, I'm just converting the labels to colors, and now I'm getting my module eigengenes. That's what ME stands for. Yeah? And I'm storing them in a variable called MEs. Let's look at this variable. It's just a data file. Okay? So I have $19 columns and 134 rows. What does it tell you? I have 134 rows. This number should be very familiar by now. What is 134? How many samples do I have? 134. For each gene, I have expression for 134 samples. For each phenotypic trait, I have 134 samples, right? So each of these eigengenes, they represent one module. I have 19 different modules, the different colors. Module yellow, module pink, module turquoise, module brown. So the genes are clustered. All the 3,600 genes I have, I'm clustered into these different modules. But I cannot correlate an entire module to my phenotypic trait. So I'm calculating my eigenvector, my eigengene first. And this is the eigengene. And this eigengene is one column, yeah, ME9 is one column times 134 rows. If I can now correlate each eigengene to each phenotypic trait. You look a bit puzzled. Do you understand what I'm saying? I will show you an example. Okay. I will store, I will call it sample eigen gene. I have my data frame of eigen genes. What was it? MEs. I will only look at the first one. How many values are in my sample eigen gene now? It's a vector. How many variables do I have in my vector? How many? Who says 1? Who says 19? Who says 134? Yeah? So I'm looking now. Just to demonstrate it to you. 134. I have 134 values. Now, where do I have my trait data? All traits, right? I will copy it. It's the weight of the mouse. Yeah. But this is essential. 5 more minutes, okay? And then we go into break. Now, I have my traits. I have my data frame of traits. It was more than 300 different measurements for each trait. I cut it down so it corresponds to the samples that I have in my my gene expression file. Okay? So I have 134 measurements in my cut down data for my traits. I have 134 length vector, which is my eigen gene, corresponding to a specific module that was calculated using WGCNA. So I have 2 vectors of same length, right? Each one has 134 variables in them. Now I can correlate them. Function core, sample eigen gene to my first trait. Okay, so I now correlated using Pearson correlation my sample eigen gene, my first eigen gene, to my first trait. Wait, I get a correlation coefficient of negative 0.01. Is this significant? That's correlation coefficient. No, not at all. That means in the module that I chose, there are no genes that somehow are regulating the weight of the mouse, which I measured in the liver of the mouse. The expression data that I got from the liver of the mouse doesn't correlate to the weight of the mouse. And that's the idea of WGCNA. After you construct your module, you calculate your module eigengenes, and then you correlate those module eigengenes to your phenotypic traits. When you get a high correlation, may it be positive or negative, you then look into the specific module, look at the genes that you have in this module, and try to identify genes that could regulate the trait that you're looking at. Now we can go on to the break. Thank you.